to meet Dr. Michael Press from our organization, who is an MD, PhD, and he's on our medical advisory board. So he is the gentleman that is the forefront of actually finding a cure for breast cancer. So the reason why all of you are here is to make sure that he gets to do what he does on a daily basis.
TheGreenGirls.com.
In 1987, Dennis Slayman at UCLA identified a molecular genetic alteration that was in approximately 25% of breast cancers, known as the human epidermal growth factor receptor type 2, or HER2, or NU, and this gene was increased in copy number, so it's called gene amplification. Now, as many of you probably already know, breast cancer is a genetic disease. About 10% of women have breast cancer based on the inheritance of a mutated gene, and the vast majority of women have mutations that develop in their breast tissue during their lifetime. And one of the challenges in the lab is to identify which genes in cancer, especially breast cancer, are altered, and then of those hundreds of genes, probably thousands of genes that are changed, which of them play an important role in the growth or maintenance of those cancers. And then the goal is, after you've identified those genes, to say how do they work, and can we step in and block that function in a way that will change the outcome of that disease. So this alteration that Slayman first identified in '87 was one that we were very excited about. I stopped the work that I was doing in my laboratory, then in Chicago. I came to UCLA and worked with him for a year in his laboratory on a sabbatical leave from my job at the University of Chicago. We worked together for a year in order to better evaluate this alteration, how it functioned in breast cancer, and what its potential role could be. The good news of this story, as many of you I think know, is that this gene coded for a receptor on the surface of the cell, which allowed antibodies to be made to the part of that protein that stuck outside of the cell. One of those antibodies became humanized and is now known as the drug Herceptin, or trastuzumab. And in a series of clinical trials, this drug was shown to be effective first in women who had disease that had spread in their body, and then subsequently in women who had early disease. Now, this represents really a big operation across many laboratories. Not only the discovery activity at the laboratory site, but then characterizing these genetic alterations in the lab, and then finally developing a therapeutic that can block it and testing it in patients. So the ultimate goal is to get it into treatment as early as possible. In August of 2000 through March of 2004, my laboratory at USC and a satellite laboratory in Basel, Switzerland, began the screening of women's breast cancers who were candidates for entry into a trial of early breast cancer. We screened over 10,000 cases, 10,000 breast cancers, in order to identify over 3,222 women whose breast cancers had this alteration and were entered in the trial. The analysis of that clinical trial is complete, and the use of trastuzumab, in addition to their chemotherapy, which was standard of care, resulted in a reduction in the recurrence of their disease by fully 50%, or slightly more than 50%. It's not only in a clinical trial from our group, the Cancer International Research Group, but there are 3 other two American groups and 1 international group that have shown similar findings. So this was very exciting for us. We now have another trial that's starting up that's going to be of a similar size, where the goal will be to test this biologic agent. This is a humanized antibody to a molecular genetic alteration. There's now another antibody that blocks the growth of blood vessels. We'd like to see if these 2 biologic agents together can further improve the outcome of women who have breast cancer. So this is a very active process that's international across many laboratories. I think you can be optimistic. There are literally thousands of genes that are known to be altered. There's an ongoing process to identify which genes are important in the disease process, and then more specifically, which of those that are important can we block their activity, and which of those can we get into clinical trials. So that's just a very brief summary of the kind of work that's being done in my laboratory, in Slayman's laboratory at UCLA, and at many other laboratories across the United States and internationally throughout the world.
TheGreenGirls.com